shuttling of the pyruvate to the mitochondrial matrix, and then the oxidation of pyruvate into your acetyl coenzyme A. The citric acid cycle is also going to serve as the final common pathway for the oxidation of fatty acids, and this is going to occur through the beta oxidation process. And the beta oxidation of fatty acids, as shown over here, is going to lead to the production of acetyl coenzyme A, which can then be transported to the mitochondrial matrix, or which can be fed into the citric acid cycle. And last but not least, the citric acid cycle is useful for serving as the common pathway for the oxidation of various amino acids, because that's eventually going to lead to the production of acetyl coenzyme A, which again is the necessary fuel for initiating the citric acid cycle, or the tricarboxylic acid cycle. And as this cycle happens, what's going to happen is you're going to generate reducing equivalents, specifically the NADH and the FADH2. These reducing equivalents will be necessary for fueling the next step of energy production, which is oxidative phosphorylation. And you can see these reducing equivalents or intermediates being consumed over here. And last but not least, the citric acid cycle is useful for providing various biosynthetic intermediates. For example, alpha ketoglutarate is useful for producing glutamate, which can then be used to produce glutamine. Regarding the location of the citric acid cycle, is going to take place in the mitochondrial matrix, and the only exception to this rule is the enzyme called the succinate dehydrogenase, because the succinate dehydrogenase is actually embedded in the inner mitochondrial membrane rather than the mitochondrial matrix. And this is because the succinate dehydrogenase serves as the complex 2 of the electron transport chain. Alright, and now let's talk about some general overview of this cycle. So, what ends up happening is that one acetyl coenzyme A will combine with oxaloacetate in order to produce your citrate. So the citrate is going to be a 6-carbon entity, and this is because the acetyl coenzyme A has 2 carbons and an oxaloacetate has 4 carbons. And after this happens, you're going to have 2 carbons that end up being released as the CO2 molecule. One over here and one over here. And so you're going to have to subtract 2 from 6, and that's going to leave you with 4 carbons remaining. And what's going to happen to those 4 carbons? Well, you get your oxaloacetate again, because remember that oxaloacetate has 4 carbons. And this is why this is known as the citric acid cycle rather than the pathway. It's a cycle that continuously produces intermediates that are essential for continuous generation of energy, so long as you're able to obtain acetyl coenzyme A. Alright, now let's break down the cycle step by step. So, the first step is to form citrate, and this is going to happen through the enzyme citrate synthase. The reaction involved is your acetyl coenzyme A combining with oxaloacetate and water in order to produce your citrate, plus your coenzyme A SH molecule. molecule. And then the key points behind this step is that this is an irreversible rate-limiting step, and this step is going to be inhibited by the ATP molecule, the NADH, the citrate molecule, and the succinyl coenzyme A. Notice how the molecules I mentioned, like the succinyl coenzyme A, the citrate, the NADH, and the ATP that is eventually produced in oxidative phosphorylation right over here, they provide negative feedback to this important reaction. And this is a classic example of negative feedback from the generated product, because if you produce a lot of ATP, that means you don't have to make the cycle continue to operate, as you don't need any more ATP in your body system. So that's how feedback mechanism works for this particular step. Let's progress to the next step, which is the isomerization of citrate. So in this step, we have the enzyme called aconitase, and the aconitase will convert citrate to cis aconitate, and then eventually to isocitrate. And this is through the process of isomerization. The key points behind this is that you're going to have the cis aconitate intermediate. And then another important concept is that this molecule called fluoroacetate or fluor will inhibit the aconitase. And what's going to then happen is the tricarboxylic acid cycle will be arrested. That is why fluoroacetate is toxic. Let's move on to the next step, which is the first oxidative decarboxylation step. So in this step, you have the enzyme called isocitrate dehydrogenase, and it's going to convert your isocitrate into your alpha-ketoglutarate. By doing so, it's also going to generate one CO2 molecule. Another special thing about this step is that this is where you're going to generate your first NADH molecule. And remember that NADH is an important intermediate for fueling oxidative phosphorylation, as shown over here. So this step is important because it is activated by your ADP molecule and your calcium ion, but it will be inhibited by your ATP and NADH molecules. It is also the major regulatory step of the cycle, so keep that in mind. As we go to the next step, we're going to have a second oxidative decarboxylation, where this time we have the alpha-ketoglutarate dehydrogenase complex that is going to facilitate the conversion of your alpha-ketoglutarate into your succinyl coenzyme A. Again, this step is special because another CO2 molecule is going to be generated. What is also special about this particular step is that various cofactor molecules are involved. So your TPP, or vitamin B1, as shown over here, will help the enzyme to facilitate this conversion. Also your lipoic acid, your coenzyme A, like vitamin B5, FAD molecule, or vitamin B2, or NAD+, or vitamin B3. All of these will help to catalyze assist the alpha-ketoglutarate dehydrogenase to catalyze the conversion of alpha-ketoglutarate to succinyl coenzyme A. On the other hand, this step is inhibited by your NADH molecule and your succinyl coenzyme A through the product-based feedback or product-based negative feedback mechanism. And remember, once again, that calcium is going to help activate this step. So factors I mentioned about for this step are similar to the ones associated with the pyruvate dehydrogenase enzyme. On the other hand, the arsenic is going to inhibit the lipoic acid, which again is an important cofactor for this step. And then for people with thiamine deficiency, this step is going to be somewhat inhibited or reduced, and ultimately this is going to lead to decreased citric acid psychoactivity. So that is why vitamins are important. So be sure to get your vitamins. Alright, let's go on to the next step, which will involve substrate level phosphorylation. So this time, once we have our succinyl coenzyme A, we have our enzyme called succinyl coenzyme A synthetase. And this enzyme is going to convert your succinyl coenzyme A into succinate. Now this time, the product that will be generated is your GTP molecule, or guanosine triphosphate molecule. Then again, the GTP can eventually be utilized to form ATP. And another important thing about this step is that this is the only substrate level phosphorylation involved in the TCA cycle. And you can see the phosphorylation step labeled here. So once we produce our succinate, it is time to oxidize your succinate into your fumarate. And this is going to happen through the enzyme complex called the succinate dehydrogenase, which again is complex 2 of the electron transport chain, or oxidative phosphorylation. And so this is going to lead to the production of fumarate from the succinate. And you're also going to produce FADH2, which can be used to fuel oxidative phosphorylation. On the other hand, something that can inhibit this step is this thing called a malonate, which happens to be a competitive inhibitor of this enzyme. Alright, now that we have our fumarate, the next step is to conduct hydration of fumarate in order to produce your malate. And this is going to happen through the enzyme called fumarase. And the fumarase enzyme will basically utilize the hydration of fumarate and produce malate, which is why a water is consumed over here. Hence the term hydration. So once we have generated our malate, now it's time to recreate the very essential component of the TCA cycle, which is oxaloacetate. This is going to happen through the enzyme malate dehydrogenase. So remember that malate dehydrogenase will convert malate into your oxaloacetate, and by doing so, it's going to generate another NADH molecule. And remember, this NADH molecule can be used to fuel the oxidative phosphorylation process. So once we have that, we return to the starting point of the citric acid cycle, or the first step, as we elaborated on earlier. And one more thing to note about this malate dehydrogenase step is that the reaction is unfavorable, but it is nevertheless pulled forward by the action of the citrate synthase, as the citrate synthase will consume oxaloacetate to form citrate once we have the acetyl coenzyme A present. Now that we have gone through each step of the citric acid cycle, let's just briefly go over the energy yield for this cycle. So for every molecule of acetyl coenzyme A that you consume for the citric acid cycle, you're going to produce 3 NADH molecules. 1, 2, 3. produce about 7.5 ATP molecules. You're also going to produce one FADH2 molecule, as shown over here, and that's going to produce about 1.5 ATP molecule. And finally, you're going to produce one GTP molecule, as shown in this step, and that's going to produce about one ATP molecule. So in total, the TCA cycle itself can help to generate 10 ATP molecules, and that is why it is a very important cycle for energy production. Additionally, I want to re-emphasize the key regulatory enzymes in this cycle. So the citrate synthase is one of them, along with the isocitrate dehydrogenase and the alpha-ketoglutarate dehydrogenase, and these will be regulated mostly through the product-based negative feedback mechanism that we talked about earlier. And then if the human body is in a high energy state, that's going to mean that there's a lot of ATP produced and a lot of NADH produced. And through negative feedback, that's going to inhibit the activity of the TCA cycle. But in low energy state, where we have increased ADP molecule and increased calcium ions, that's going to help to drive the TCA cycle forward so that we can generate more ATP molecule. Alright, let's now talk about the amphibolic nature of the TCA cycle. So, as I kind of alluded to earlier, the TCA cycle is both catabolic and anabolic. So, it is anabolic because you can form various intermediates like the citrate, your alpha-ketoglutarate, succinyl coenzyme A, and oxaloacetate. These intermediates are useful for producing other important molecules in our body. So for citrate, it is essential for producing your fatty acids and cholesterol. For your alpha-ketoglutarate, this is necessary for producing glutamate and ultimately neurotransmitters. For your succinyl coenzyme A, it is one of the first components needed for producing your heme, also known as heme synthesis, which again is an important component of the red blood cell. And then your oxaloacetate is necessary for producing aspartate and ultimately fueling gluconeogenesis, i.e. the production of glucose. So you can see the important roles these intermediates play in helping to maintain our body functions, biochemically speaking. Let's also talk about the nature or the important aspects of the anaplerotic reactions, which is also the reactions necessary for refilling this cycle. So, the most important one involves the use of pyruvate carboxylase, as shown over here. So the pyruvate carboxylase is actually going to help convert your pyruvate into your oxaloacetate. And this particular step is going to require your biotin or vitamin B7. Vitamin B7 and your ATP, as shown over here. And interestingly, this step is going to be activated by your acetylcoenzyme A, which makes sense because if you have your acetylcoenzyme A waiting to be fed into the TCA cycle, you need your oxaloacetate in order to initiate such a process. And so acetylcoenzyme A can basically tell the pyruvate carboxylase to hurry up and produce more oxaloacetate from your pyruvate so that it can basically join the oxaloacetate and through the help of citrate synthase produce your citric molecule. It does help to fuel the TCA cycle. And so this step is clinically relevant because it is essential during fasting and gluconeogenesis. And I say that because oxaloacetate, it can actually be shuttled back to the cytoplasm and eventually be utilized for gluconeogenesis or the production of glucose. So there's some other clinical correlates that I want to highlight. So unlike many other cells, there's no TCA or citric acid cycles happening in red blood cells because once again, this cycle happens in a mitochondrial matrix and red blood cells do not have mitochondria. In situations of hypoxia or low delivery of oxygen to tissues, there's going to be decreased electron transport chain activity because oxygen is necessary for this step. And so, as a result, you're going to have less consumption of NADH and ultimately increased buildup or increased presence of NADH molecule. And once again, through the feedback mechanism, the built-up NADH molecule is going to inhibit the enzymes we talked about earlier, like the citrate synthase, isocitrate dehydrogenase, or alpha-ketoglutarate dehydrogenase. And what's going to happen next? Well, the TCA cycle is going to slow down, or in other words, kind of be inhibited. Another clinical correlate to highlight is thiamine deficiency. So remember how thiamine is essential for the activity of alpha-ketoglutarate dehydrogenase. So decreased activity of this enzyme is going to kind of slow down the TCA cycle, or at the very least, decrease the production of succinyl-coenzyme A. And so the inhibition of this cycle is then going to lead to the metabolic pathways having to rely on the production of lactate. And as a result of this, there's going to be lactic acidosis. Thiamine deficiency is also known to induce neurologic symptoms. And then lastly, we have arsenic poisoning. And this is problematic because arsenic inhibits the lipoic acid, which remember we talked about how lipoic acid is an important cofactor for the alpha-ketoglutarate dehydrogenase complex, as shown over here. And so this is going to also inhibit the TCA cycle, and it cannot actually mimic the thiamine deficiency, since thiamine deficiency and arsenic poisoning will lead to this step being compromised. So those are the clinical correlates to keep in mind. And some big picture summaries that I want to highlight are that the TCA cycle supplies your NADH and FADH molecules that can be consumed in the electron transport chain to produce your ATP molecules. And then another important thing about this cycle is that oxygen is indirectly required, because in order for these intermediates to be consumed in the oxidative phosphorylation step, you need oxygen. So that is why oxygen is indirectly required. And what's special about this cycle is that it serves as the central metabolic hub for connecting carbohydrate, fat, and protein metabolism. And that is why it is important to know the basic principles and the clinical correlates associated with this step or with this cycle. So that'll be it for today's video. this video to be educational and also tingly for those of you who love ASMR content. I hope to produce more ASMR videos like this for you in the future. In the meantime, feel free to comment down below any clarifications that need to be made or any corrections that have to be addressed, because I am not always accurate with my explanations, or sometimes not even comprehensible. So feel free to comment down anything else that needs to be mentioned. And otherwise, like and subscribe for more ASMR